What advice would you give aspiring young scientists who are embarking on their scientific journeys?
Don't listen to advice.
Okay.
I think there's a little bit of a variant of that question, and that variant is, how do you get a Nobel Prize? And I have a very definite answer to that, and that is, I have not a clue of how to do it, and I don't think hardly anyone else has either, because if we knew, if the Nobel laureates knew how to get a Nobel Prize, it is such a wonderful party. It's so great that we would have gotten our second and third by now.
Welcome to the 71st episode of the Struggling Scientist podcast. We are a podcast by scientists, for scientists, anybody science adjacent, and perhaps even hobbyists. My name is Susanna, and I'm here with my co-host Jaron.
Hi.
For our 70th episode, we wanted to do something special. So we invited Nobel Prize laureate Professor Martin Chalfie to be our guest. His groundbreaking work with the green fluorescent protein, also known as GFP, has revolutionized the way we understand the inner workings of living organisms. And since he was kind enough to talk with us for over an hour, this episode will be split into two parts. In episode 70, we ask him all of our questions about his scientific career, how we almost decided he was not cut out to be a scientist, and of course his discovery about using GFP as a marker. In episode 71, we'll talk about the impact winning a Nobel Prize has on your life and how he missed the announcement of his own Nobel Prize. He has a lot of amazing stories to tell, so let's start.
So in episode 70, we started our interview with Nobel laureate Professor Martin Chalfie. We talked about his scientific career and all the struggles that came with it. How he almost decided he was not cut out for a scientific career after months of failed experiments, and about all the factors that led up to his scientific discovery of GFP as a marker. And now we'll continue our interview with Professor Martin Chalfie. So following the publication of your landmark paper, how was it actually received? Did you have already the feeling that this was going to be potentially Nobel Prize worthy or not?
I don't think I thought about it as Nobel Prize or prize worthy. I just thought it was going to be a very useful tool. And I was a little bit surprised. There were a couple of reactions that I was not really, I think, prepared for. And so going back even to that first seminar, I was exceptionally excited about GFP as a potential thing, and nobody else in the room was. And I've been trying to think about, why weren't other people excited? Was it because most of the people in the room were neurobiologists? Or was it because they all worked on animals that weren't transparent? And I suspect that was the main thing. Oh, here's some interesting fact. But I'm working on a transparent animal, and someone's just given me a light bulb. I'm really excited. But when our experiments started working, I think basically everybody in my department knew about the work. Nobody wanted to use it. No one came to me and said, "Can we use it?" Except Tula, my wife, wanted to use it for her own experiments and saw the importance of it immediately. It wasn't me. I mean, she said, yeah, she got excited. She used it as a tool. But the rest of the people didn't. And so it was a little slow. I think C. elegans people, once we showed, we actually, the official paper came out in in February. But in October of 1993, so before the paper, we had a one-page description of GFP for the what we call the Worm Breeders Gazette, which is sort of the C. elegans newsletter. And we were sending out samples all over the place because people were reading this. So that was really the first public announcement of it. And we were sending things away to the C. elegans people. And then more and more people would start calling us and say, I've heard about this. interestingly, they usually started this phone call by saying, my postdoc or my graduate student tells me you have this new technique. What is it, and should we get it? And then they'd agree. But and that continued after the paper was published. But people picked, I think it really was graduate students and postdocs that pushed the idea, here's a new technology, we can go and use this. This is exciting. There were plenty of other people that did as well. I don't want to disparage anything. There was one person who called me up and said, has anyone done, as many people would call, has anyone put GFP yet in X, whatever they were studying? And I'd say, no. Most of them say, great, I want it. Every once in a while, you get somebody who said, I'll wait till they do it. And I'm a little surprised at that. I also was exceptionally naive about biotech. I had this very naive idea that companies had this magic place called the Division of Research and Development, R&D. And I thought for sure that a company was going to say, this is such an obvious tool. This is so, there's going to be so many uses for this. We want to develop it. We want to make it stronger, get different colors. We want to do all the modifications. And I learned to my, frankly, horror that companies love to license things, but they didn't want to develop them. And so the development actually was done by other academic scientists. And that I found very surprising. I thought there'd be some place that had 100 chemists or molecular biologists, and they'd just go to town, and in about a couple of weeks, they have all the modifications and everything had been made. But that really didn't happen. There was one company in Denmark that was sort of set up to do something like that. But for the most part, no. And I found that a little disheartening. But, I learned.
Also, their loss, they could have made a lot of money.
Yeah.
So we're skipping ahead a few years now. GFP was already being widely used at that point for many, many practices. And then you were awarded the Nobel Prize in chemistry in 2008. How did you hear that you had won the Nobel Prize, and how did you react?
Well, the fun story is that it goes back a couple of years before the Nobel was announced. My Nobel was announced, where there had been well-meaning friends who had said to me, GFP is pretty important. We think it might get a Nobel Prize. Now, if you're an idiot and somebody says that to you, you get all pumped up and excited, and you can't sleep the night before that supposed announcement is going to be done. And so you lose a night's sleep and they don't call. So I was a fool. I had stayed up because I had gotten excited about and wondered, I got to be awake for the phone call. That's really important. So in 2008, on the Tuesday night before the announcement of the chemistry prize, I got an email from a student in China. And in the email, he asked for a copy of the PDF, the science paper. And in his email, he said, tomorrow is going to be the chemistry prize, and my friends and I really hope that it's going to go to Shimomura and Chen. And I looked and my name's not there. And I read the thing several times, and I write him back, and I say, here's the PDF. They can have 3 people. And what about Douglas Prasher? Or, there's other people, or Tula Hazelwood, my wife. And he writes back, and he says, no, actually, because of his Chinese descent, we're really hoping Roger Chen gets it. I was out of the picture entirely. And I went to, I thought this was hilarious. And so I showed the emails to my wife, who I've always already said is very important in getting GFP to be useful. And Tula looked at me, remembering the nights that I had gone sleepless and there had not been a phone call, and just looked at me and said, Marty, GFP has been a very, very useful tool. It is a wonderful thing. Many people use it. But I have to tell you the truth. It is never going to get a Nobel Prize. So just go to sleep. Which I did. And we live in an apartment in New York City. And the phone was in the kitchen. There are 2 closed doors between our bedroom and where the phone was. And through some craziness, I had already I had done something to the phone, so the ring was quite soft anyway. Slept through the entire phone call. Missed it. Absolutely. Woke up at around 6:30, 6:20 in the morning and realized that there must have already been the Nobel announcement. So I happened to have my laptop near the bed, and I opened it up and I went to the nobelprize.org website to see who the winners were. It's a shock when you see your own name in that list. And I was very surprised. I woke up Tula and I said, look. I said, no. I said, it happened. And she immediately jumped out of bed. Said, oh, no. And because she thought what I meant was that our teenage daughter had overslept and had not high school, and that we had to get her to high school soon. I said, no, she doesn't have to wake up for another 10 minutes or so. No, look, here. I showed her the thing. And then there was more jumping around. and stuff. So that was so I missed the announcement. But it was a wonderful day.
I can imagine, yeah.
It's and it's a wonderful party, wonderful celebration. It's the Nobel is, I think, unique in that an entire country takes off a week to celebrate academic achievement, and that's pretty remarkable. And so. It's quite wonderful. I recommend it to everybody. If you can get one, please do. You're going to enjoy it.
Yeah. I imagine your life must have gotten quite, even busier with all the interviews and such with the announcement, after the announcement was made about the Nobel Prize. Looking back, were there moments where you could just sort of appreciate the fact that you had just become a Nobel laureate, or was it all so busy and hectic that you couldn't really take the time to appreciate it?
No, you get a chance to slow down and as I've sort of indicated, I have people at home, my daughter, my wife, who make sure I know when when my head's swelling too much, and you get a check on reality. It's a surprise. And a very enjoyable one, and unimaginable consequences of having this happen. You get invited to podcasts.
Yeah. Yes.
You get invited to talk to students. And you get I have been fortunate to be asked and actually elected as the president of the Society for Developmental Biology, and just finishing up as past president now of the American Society for Cell Biology. And those opportunities to serve, I don't think would have come my way without the visibility that the Nobel has given me. I don't think my involvement with the Committee on Human Rights would have been enabled had I not been able to, that I didn't have this visibility that adds to our work, if we can say that. We sometimes try to get in touch with different foreign embassies to talk about cases that we're concerned about. And it's a little easier to get through the door if we say, well, the chair of the committee is coming, he has a Nobel Prize. And I have had people say, you're the second Nobel Prize winner we've talked to. And so if it gets us in the door, if it gets people to listen, I think that's wonderful. It's an appropriate use of the visibility. There are things that I find surprising. There are some people that want to take advantage. You have to be a little careful. People want to use the Nobel. So you have to be a little vigilant. But it's growing up. You learn a few more things as you go through this. But I think, by and large, it's been wonderful. I think one of the really fantastic things has been being able to work and talk with as many students as we do, as I've been able to have an opportunity to. We have a course here for undergraduates. We have a series of courses we call the Core at Columbia. And starting about 12, 15 years ago, one of the core courses was in science. And for the last 4 or 5 years, I've been asked to talk to all the freshmen undergraduates in the course. And it's really interesting. It's really fascinating to see their excitement and what they're interested in. And so the Nobel has given me an opportunity to reach out. There's been a little bit of a push to reach out too, because you feel, now I should be doing something. I should be contributing more than maybe I have in the past. So it's not only that you have the opportunity, but you also feel a little bit of an obligation, a thank you to everybody about what's really been a wonderful career.
It sounds like the Nobel Prize has impacted your life quite a bit. Has it also influenced the direction of your scientific endeavors at all?
Not really. As I said before we worked with GFP, I was interested in nerve cell development and mechanosensation, genetics of both. And we continue to work on that. We work on different aspects of it, but the prize itself has not changed anything. I like to say, we did the experiments in 92, published them in 94, but the prize wasn't until 2008. I wasn't sitting on my hands waiting for the prize. Sure. I was doing those other experiments, and we still do those other experiments. And so it hasn't. That's not to say that there may be times to think about, what other directions should this be going in? I think one of the things, because there are a lot of demands on one's time, that it's not so much that the experiments go in a different direction, as much as there are suddenly many more opportunities. And so what are your priorities? And I'm still trying to come up with that and to think about that. I'm actually technically on sabbatical this year, and I'm going to spend the spring, I think, really thinking about how I want to proportion things, catch up on my reading, and talk to people about other experiments and so on. But I think that it does put a spotlight on you, and that spotlight can either be overwhelming. Some people ignore it entirely. I think it's a variety of approaches, and also this doesn't come when you're so young all the time. And so that part of the thinking, I don't know if it's the Nobel or thinking about where do I want the lab to go in the next 5 years? What do I want to accomplish? So we'll see what happens.
So maybe to touch on a little bit of what you just already sort of alluded to. So I think many researchers, maybe not so secretly, dream one day of winning the Nobel Prize, but it's what you sort of already alluded to. It's not all upside. There also is some sort of hidden cost or opportunity costs that come with it, I guess, that many researchers probably don't think about, such as their time, the opportunities that they do need to say no to or yes to. Are there any other sort of hidden costs that you didn't expect when you got the Nobel Prize, I guess, that came up?
No, one other advantage to just add to it is I've had an opportunity to travel to places I've never been before. And that opportunity has been wonderful. And it continues. And I enjoy that very much, meeting people all over the world. But yes, you have to judge your time. I did allude to the fact that there are sometimes requests that are a little too good. You have to be a little bit more careful about in terms of what you're doing. But by and large, not really any bad parts to it. I mean, unless I like to complain as much as anyone else, but this is a very hard thing to complain about. You sort of no longer have a license to complain, except maybe privately. But it is very nice. There are a couple things that people. I think that when we think about the Nobel, or maybe I'm talking about myself before, we put it on a pedestal. It is what we consider sort of an ultimate accolade of what we could aspire to. And because of that, I feel that we put all of our hopes and dreams on that Nobel Laureate. So the way I like to put it is that I think people feel that there are two long lines of people outside my laboratory door. One line is a bunch of people with bags of money that are coming to me and saying, "Oh, I really want to support your research. Here's another $1 million. Set up this or do this." And there's nobody like that outside my door. No, they don't come and throw money at you. My experience, many experiences of people that have gotten Nobel. Maybe the grant reviews are a little harder. What have you done lately, sort of thing. And the other thing is that they think, oh, there must also be a long line of people that are clamoring to come to your lab. I haven't seen that either. I don't know if some people are threatened, think that they will be judged harshly or whatever. But all the things, so these are the things, people always want to have people in their labs, and they want to have funding, and they want to have their papers accepted immediately, all these wonderful things. And so I think people imagine that must happen to the Nobel laureates, because if it doesn't happen to them, we're all doomed.
Yes.
None of us are in any good shape. If everything perfect doesn't happen for them, then it's not going to happen for anybody. But that's not really true. But no, those things don't happen. You become quite normal after the prize, except you do get wonderful invitations to interact with people.
Okay. Now, we also found on your Wikipedia page that you recently this year received an honorary degree in physics from the University of Parma. As far as we could find, you have not attended this university, neither worked there. How does something like this happen? And secondly, do opportunities like that now just happen often, or is this a special one? Or
so this is a special one, but yes, these opportunities do come up periodically. I think I have something on the order of about 8 honorary degrees from various places. Some of them have, one or two of them. One, Middlebury College, is one that stands out in my mind. I had a former graduate student who was a professor there. He invited me to come and talk, and I gave a talk there, and had a wonderful conversation with the president of the college, and just had a wonderful visit. And then it was very surprising that 6-8 months later, he said, "Well, you're getting an honorary degree." So I must not have annoyed too many people, and so that happened. The Cristiano Vapiani at the University of Parma was the person who initiated this. One of the absolutely wonderful experiences I have had is I got an invitation soon after the Nobel was announced, or in that year, to attend the European Biophysics meeting, which was in Genoa. And Cristiano was there, and other people, and particularly a man named Alberto Diaspora. And these are all people that worked with Albi. Albi is one of the great people in the world, unbelievably nice guy. And I have a relationship with the Italian Institute of Technology where he is, and it's in Genoa. And I go there every year because we have a science advisory board meeting. And just that entire group of people that worked on microscopy and fluorescence and stuff have been unbelievably kind to me. And Cristiano arranged for this, or nominated me for this honorary degree in physics, which I don't deserve, and arranged for that. And it was a very nice ceremony. I have no idea who put that on the Wikipedia page. I don't put any. I used to read the Wikipedia page to see how many mistakes there were, because I could. This sounds horrible, but when people would introduce me, because of the mistakes in the Wikipedia page, I could tell where they got their information. So it was sort of a little joke on that I enjoyed, but yes, I have an honorary degree in physics now.
That's so much fun.
It's astonishing.
So I might want to touch on just a little bit more on GFP very quickly, if that's okay.
Sure.
So GFP and its related proteins are now being sort of widely used in research. Did you expect it to develop as far as it has? And what development are you most impressed by or proud of when it comes to GFP so far?
So the answer to that is I don't think anyone can imagine all the wonderful applications that people can come up with. I have had several over the years that have been my favorites of this. I don't think there's any one. There are results. I think the discovery of phase-separated particles is Tony Hyman and Cliff Brangwyn. I think it was an astonishing discovery to be able to visualize that in cells, in C. elegans. I think was a wonderful use of GFP. I have always liked the idea of GFP as a marker for TNT that leaks out of landmines as a way of detecting landmines, but still not at a stage, I think, where it's as useful as it could be. You don't want to have any false negatives. But there, one result that I particularly was intrigued about, Jeff Waldo at Los Alamos Labs did a really interesting experiment. The logic behind it is just wonderful. He was interested in monitoring protein folding. Now, one of the problems of trying to grow up a lot of protein in E. coli, for example, is that the protein will be made, the hydrophobic regions will glom together, you get a precipitate in the cell. You don't get the properly folded protein. And these aggregates are a problem because then you have to solubilize them, hopefully allow the thing to reform. And so Jeff did this wonderful experiment where he took a protein he was interested in, and he then added the coding sequence for GFP. So he made a fusion protein of these two things. And then once he did that, of course it didn't. What happened was the protein didn't fold up appropriately. GFP would then not fold up appropriately. And so he reasoned if he mutated the original protein, maybe he would be able to find some combination of mutations that would allow the original protein to fold correctly. If it did, then the GFP could fold correctly and you'd see fluorescence. And so he basically made a library of all the different variants he could come up with. He mutagenized the DNA and then just put them in E. coli and look for the E. coli that were green, because those are the ones where the protein had to fold up first before the GFP folded up and produced a fluorescent product. So he had a wonderful way of monitoring protein folding. And I thought that was brilliant. I thought that's just a fantastic idea. I think that Lynn Regan's work showing that you could cut GFP in half, put the two halves on things that will come together and reconstitute the GFP is astonishing. And that these two component systems. I think all the work started by Roger Chen of first using FRET, and then as people use single molecule methods to bring things together to detect calcium and other molecular monitors. These are all wonderful ideas. So there's no one idea. They're great results. They're wonderful ideas. And these go way beyond anything I would have imagined. And so it's wonderful. But virtually every discovery leads to every basic discovery leads to more basic discoveries and a lot more applications and everything. Look at the laser. When Charles Towns made the laser, nobody came up to him and said, we want to make CDs or DVDs, or we work in a grocery store, we want to have something that'll help us check out the food faster, or I'm a surgeon, I want to do surgery, I need a really detailed way of being able to do the surgery. These all came after, and they're quite remarkable. Look at all the stuff that's coming now out of CRISPR-Cas9. The discovery, the initial things come out, and then it explodes in many directions, getting new techniques, but also new knowledge. Laser, going back to that, spaces, ultra cold, gravity waves, laser tweezers, all sorts of wonderful things that have led in their turns to other discoveries. So, no, I didn't imagine most of these things, but I'm very happy for them. I'm very happy that they have happened.
Yes. I think also great progress for science. Pretty amazing. What advice would you give aspiring young scientists who are embarking on their scientific journeys, especially in the context of exploring new frontiers in biomedical sciences?
Don't listen to advice.
Okay.
I think that, there's a little bit of a variant of that question, and that variant is, how do you get a Nobel Prize? And I have a very definite answer to that, and that is, I have not a clue of how to do it. And I don't think hardly anyone else has either, because if the Nobel laureates knew how to get a Nobel Prize, it is such a wonderful party. It's so great that we would have gotten our second and third by now. But none of us, or hardly any of us, Fred Sanger being an exception, we don't have a second Nobel Prize. We have no idea how to do it. So I'm going to take this opportunity to rail about something that I think we tell talented students at all stages. We say, follow your passion. Okay, I hate the phrase. I'm going to tell you why I hate it. If someone had said it to me, I'll tell you exactly what I would have said to them. I would have said, I don't know what my passion is. And I'm really afraid I'm going to take the wrong path. Tell me what my passion is, because I don't want to make a mistake. So I think all we do by saying, follow your passion, is scare the hell out of people. So I think what we really mean to say is, it really doesn't make any difference. Be passionate. Be excited about what you're doing. Go into it totally. Don't hold yourself back. But then again, almost all advice, you're not telling it to other people. You're basically telling it to your younger self. So that should be taken with a grain of salt on this. I think, but I think being involved is important. And I do wish I had been more involved earlier in my life. But I can't complain.
Still plenty of time. Well, thank you so much for joining us on our podcast today and answering our questions. Is there anything we have not asked you that you would still like to mention?
No, I think we've covered quite a lot.
Okay. Well, then it was an honor to talk to you, and we hope many scientists will be inspired by your story.
Yes.
Well, I had a wonderful time. Thank you both for doing this.
Yes.
Take care.
For our listeners, if you have any questions, suggestions, or comments, you can reach out to us via our website, thestrugglingscientist.com. You can also check out our website to sign up for the awesome Journal of Struggling Scientists, aka our newsletter. And if you have enjoyed this episode, please leave us a rating on your favorite podcast listening platform, as it helps us grow and reach out to more struggling scientists out there. You can also follow us on social media. Jaron, which ones are those again?
X, Instagram, LinkedIn, Facebook, and YouTube.
Yes. Thank you so much for listening, and we hope to see you again next time. Bye.
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 Yes.